Hi, everyone. This is Tara Coleman, the Executive Director of Children's Leukemia Research Association. In preparation for our 60th anniversary, we are connecting with our grant recipients to talk about their amazing work and progress on research. I'm so honored today to introduce Dr. Kimberly Stegmaier. She is a professor of pediatrics at Harvard Medical School, the Ted Williams Investigator and Vice Chair of Pediatric Oncology Research at the Dana-Farber Cancer Institute, and the co-director of the Pediatric Hematologic Malignancies Program at Dana-Farber and Boston Children's Hospital. She is also an institute member of the Broad Institute of Harvard and MIT. She received her Bachelor of Science degree from Duke University and her medical degree from Harvard Medical School. She completed a residency at Boston Children's Hospital and Pediatric Hematology Oncology Fellowship at Dana-Farber and Boston Children's. In 2006, she launched her own laboratory at Dana-Farber Cancer Institute. Dr. Stegmaier is on a number of important boards, including the American Association for Cancer Research, AACR, the largest cancer research foundation in the world. In October 2024, Dr. Stegmaier was appointed by President Biden to the National Cancer Advisory Board, which helps guide the director of the National Cancer Institute in setting the course for the National Cancer Research Program and Cancer Moonshot Project. Dr. Stegmaier has been receiving special research grants from CLRA since 2013. Can you tell us a little bit how your initial grant from CLRA came about and how you met our President Emeritus, Anthony Pasqua?
Sure. So, Tara, thank you for that very kind introduction. And I want to thank CLRA for all of the support over the years. It has just made a tremendous impact on our work and childhood leukemia. So thank you all so much. It takes us back to 2013 when I first met Mr. Pasqua, and I was invited to present by the Dana-Farber at the Chief Scientific Officers Forum. And this particular forum was focused on leukemia, new discoveries and new technologies. So Mr. Pasqua was at that meeting and heard me present work from my lab that was focused on using new genomic technologies to find new targets and ultimately new drugs for childhood leukemia. And so he, after hearing me present, we chatted, and he reached out to the Dana-Farber and said he was interested in learning more about my work and potentially having the Children's Leukemia Research Association support my funding. So of course, I was absolutely thrilled to hear that, of course. And then so delighted to meet Mr. Pasqua when we had our very first meeting to talk more about our work, more about his history working as the president of Children's Leukemia Research Association, and also his relationship to childhood leukemia. And I have to say, over the years, it has been just an absolute pleasure to work with him in his role formerly as the president.
That's great. Yeah. His personal connection is really what has driven our organization, this many years. We're so thankful for it and his work, and obviously for you and your work. So you had sent a progress report recently, and you discussed the focus on a particularly aggressive form of pediatric acute myeloid leukemia, also known as AML. So you mentioned problematic fusion proteins and using protein degradation to tackle these proteins. So can you explain to us a little bit in layman's terms how that system works and better understanding of the problematic proteins?
So in childhood cancer, very commonly, probably in roughly half of pediatric cancers, there's a process called a translocation, where you get breaks in chromosomes, and these breaks cause genes to come together and make what's called a fusion oncoprotein, which is a protein that comes from part of the sequence of one gene fused to part of the sequence of another gene. And then it makes this abnormal, what we call a chimera protein, or an abnormal protein that's not present at all in normal cells, and that actually initiates the cancer, and then is very important for maintaining the cancer. And so we've identified many of these fusion oncoproteins over the years across all of the types of cancers we see in children, from leukemias to solid tumors to brain tumors. So, our estimate in leukemia is somewhere around half of childhood leukemias have been identified to have one of these fusion oncoproteins. And they're what we call the driver event in that particular cancer. And so in childhood AML, there are a number of these fusion proteins that are, some of them are high risk and some of them are less high risk. And one of the types that we've been studying is called it's a complicated name term, but it's called the CBFA2T3-ETO2. Okay.
That's, I was just talking about that
It's a
today.
mouthful. It's a mouthful, fusion oncoprotein. And this particular fusion creates a very aggressive type of childhood AML that's been pretty resistant to many of the standard treatments that we use. And so, one of the, and we'll probably talk about this more later, but one of the great advances in treating childhood cancer, but also cancer more broadly, is the idea of finding more targeted drugs that target the particular changes in the cancer cells that aren't in the normal cells, right? And so in some ways, you might say that this will shorten it to CBF Gliese. So I'll use a kind of a shorter version of that long name.
Yes.
Okay, just for a placeholder. So, if you could find a drug that targeted this CBF Gliese protein, we would predict that that would be a very effective drug. And so you could say, well, okay, well, let's make a drug against it, right? So it turns out, though, that often these fusion oncoproteins in childhood cancer, the proteins that they make are actually very difficult to make drugs against. So it's generally easier to make drugs against proteins that have pockets. And then the drug can fit into that pocket, kind of like a lock and key relationship. And so, exactly. So the types of proteins that have that sort of pocket are often proteins called enzymes. So many of the targeted drugs that have been developed target enzymes. Now, the problem is the CBF Gliese protein, it's not an enzyme. And many of these cancer-promoting fusions that we see in childhood cancers are not enzymes. They're types of proteins called transcription factors, and they don't have those nice pockets. So they've been harder to make drugs against.
Wow.
So in the last several years, there's been sort of new thoughts in chemistry, right? That maybe rather than having this sort of lock and key where you have a drug that fits into a pocket and blocks how the protein works, maybe we can just what we call degrade the protein, or kind of blow the protein apart. And so this new approach in chemistry called degradation chemistry has given many of us in the field optimism that we might be able to find drugs that, again, instead of like typically inhibiting and fitting in a pocket, use the cell's own machinery for degrading proteins to degrade these fusion oncoproteins. So we've been exploring this strategy with the CBF Gliese protein. And so there are ways in the lab that are not drugs, they're artificial, what we call engineered models, to explore if you had a drug that degraded the protein, what would it do? And we have really good evidence that if we in the lab sort of with these engineered models degrade the protein, the cells in essence, they grow up, they mature, they're no longer leukemic, and they stop growing, and ultimately they die. And so we're now doing experiments with chemists and structural biologists to try to find molecules that maybe will degrade that CBF Gliese fusion protein. So it's still early, but we're really excited about this strategy for CBF Gliese, but for a very long list of proteins that are fusion oncoproteins that cause childhood leukemia. So it's a strategy in chemistry that a lot of us are really excited about.
That's it's amazing. And what's interesting and stuck with me a little bit is that it's all pediatric cancers. And
That's right.
yeah, and to
That's
be
right. It's across all the different
all of
types
them,
that we see. Yep. And,
very
roughly
interesting.
about half we think have one of these fusion oncoproteins.
Wow. And yeah, and so we always talk about this, 50, 60 years ago, we didn't have that information.
No, we didn't. No, we didn't. It's really interesting, Tara. The very first project that I worked on in med school involved the discovery of a leukemia fusion. It's called, at that point, we always have to change the names of these fusions. It used to be called TAL-AML1, and now it's called ETV6-RUNX1. Right? And it's present in about a quarter of childhood ALLs. And at the time, we just didn't appreciate, because it was back in the mid-90s, just how common these fusions were. And over the years, our field has discovered more and more of these fusions. And so it was a bit of a foreshadowing for me back in med school, the discovery of this fusion. And now
Yeah,
this is a big focus of my laboratory. So it sort of comes full circle.
it does. That's amazing. I love it. I love it. So you answered a little bit of this question. What are the most exciting or promising trends in research
Yeah,
and treatments?
yeah,
So, we talk to patients a lot, so we
right,
hear a lot about the treatments
right,
and what it side effects and long-term
right.
effects and stuff like that. So what is most promising trends in those field areas?
Yeah, no, that's a great question, Tara. And it is a very, very exciting time in science, in the lab and discovery, but also in translating therapies for children with leukemia. So, there are, on the sort of technology side, there have been a number of technologies that have helped us to better understand kind of what is behind the formation of the leukemia, and what are the kind of elements that keep that leukemia cell growing and failing to grow up or mature. And so a lot of these involve what are called genomics technologies and sequencing technologies, and the ability to manipulate genes using a strategy called CRISPR in the lab. And these new approaches, as I said, like protein degradation, single cell technologies, where we can actually study things at a single cell level, which is, back when I was in med school, was really not very possible. So it's
Yeah.
just transformed what we can do in the lab. Now, at the same time, these types of discoveries are really leading to new therapies. So one of the big areas that many of us are excited about is in the types of therapies called targeted therapies, where you're trying to target the very changes in the leukemia cell that either aren't present in normal cells, or kind of liabilities in a leukemia cell. So maybe the target is in a normal cell, but the leukemia cells care more about their target. They're more dependent on the target. In essence, it becomes an Achilles heel.
Yeah.
And so there are a whole host of new molecules that have made their way into the clinic. One is an example that we're really excited about, is a new type of targeted therapy called menin inhibitors. And so this began many years ago, about 20 years ago. This target was discovered to be important in a type of leukemia that has a fusion, right? They're called KMT2A fusions. They used to be called MLL fusions. Again, everything gets renamed over the years, but it was found that the KMT2A fusion protein interacts with menin. And that was discovered about 20 years ago. And then roughly in about 2012, an academic chemist at University of Michigan actually developed molecules that disrupt how these two proteins interact. And so they interfere with their ability to come together and interact. And this led the way then for people in the lab to start testing these compounds. And it was eventually discovered that these molecules work not just in the MLL leukemia, but in other types of leukemia that depend on menin. And so these molecules now, a number of companies have made drugs that target menin. And these molecules have been active in about a quarter of patients who have these specific gene changes in their leukemia. So the KMT2A fusions or another type of mutation called an NPM1 mutation. And the KMT2A fusions, they occur in, a number of children with both ALL and AML. And so these molecules really are extremely exciting. And I think that's one example of how science that began 20 years plus ago has led now to a drug that's in the clinic, that's active. And there was a recent FDA approval around these drugs. So this is a very exciting time, I think, in targeted therapy. And as we better understand the fusions and what the fusions interact with, many of us are very hopeful that more targeted therapies are going to come out of this sort of lab to translation strategy. Another area that everyone's been very excited about, of course, is immunotherapy for childhood ALL. This has been absolutely transformative. So, there are different types of immunotherapies. The ones that we use for B-cell leukemias target proteins on the surface of those cells. So one of the big proteins that's been targeted is called CD19. And this is only expressed on B cells. It's not expressed on any other cells in the body. So expressed on leukemias that are B-cell leukemias and B-cell lymphomas, some of them. But it is expressed on normal B cells, but we can live without our B cells. So we can help the body, support the body with the proteins that B cells make. Yeah. So it's a type of therapy that has been very successful. So there are two flavors. One are antibodies that target the CD19 positive leukemia cells, and then the other strategy is using CAR T cells, a cellular therapy. Right. And initially we were using these strategies for patients who had relapsed B-cell leukemia. And now for some patients, we're starting to bring them even into their upfront therapy. And this has just changed completely, like how we treat certainly patients with relapsed B-cell leukemias. And now we're starting to use them even in the upfront when they're newly diagnosed. So wow, super exciting times, with kind of different strategies, one being the immunotherapies and the other being the targeted therapies that aren't specifically targeting the immune system, but targeting the gene changes in the leukemia. So yeah, it's been very, very exciting time. And, these discoveries are made possible by the research in the labs, and that can't get done without support from, groups like yours. Right. So it's been instrumental in moving the field forward.
Yeah, it is very exciting. It's very exciting to hear all of this. So I'm going to skip because it's one of my further questions makes more sense now. So in one of the, we talked about this before we hit record, in one of your interviews with President Pasqua from a couple of years ago, you mentioned that your team was looking into why some pediatric patients achieve remission and some do not. Any progress on that or anything you can share about that?
Sure, yeah, that's another great question. So, it's again, like sort of the history. Yeah. Many years ago now, a frightening number of years ago when I started training, we had sort of basic information about patients and their leukemia that helped us to what we call risk stratify, to try to understand, was their leukemia a low risk leukemia or a high risk leukemia? And it was based on things like, if we look within ALL, for example, what was their age? How high was the leukemia count when they presented? Did they have leukemia also in the spinal space, the brain and the spinal space? And over the years, we've gotten better and better, both with ALL and AML, at understanding more specifically the gene changes that predispose a leukemia to being more easily treated with our current therapies or more difficult to treat with our current therapies. And I think that's been one of our big diagnostic advances, is that we've sort of been able to figure out who's at the very highest risk, and we need to give those children more intensive therapy, sometimes a bone marrow transplant, versus whose leukemia is easier to treat, and maybe we can back off on some of the more toxic types of therapies that we use to treat children with leukemia. Yeah. And this is continuing to evolve. So we have some, a new project that we're working on and starting to wrap up, where we've identified yet another risk factor that portends a slightly higher risk for certain subsets of patients based on gene changes. And so that's been a very, very big area where we've advanced in being able to predict whose leukemia is most likely to be cured, whose is harder to cure, and then changing therapies based on that information. So that's one. The other is we've gotten very good at detecting leukemia that we can't see under the microscope. And so we call this minimal residual disease. And there are different ways we can identify minimal residual disease. And so that's more information now that allows us to better predict whether a patient's likely to be cured with the current planned treatment or whether we need to intensify treatment to better help that leukemia be fully cured. So these are all like changes in, how we're treating children with leukemia that have evolved over the years. And now, of course, what we're trying to do, like those molecules that we talked about, the menin inhibitors, is for these more high-risk gene changes, can we find drugs that directly hit that gene change? And so that's to me like the next big step that many of us are working on. And I think the example of menin, or there's another gene change that involves a gene called FLT3, where we now have molecules that actually inhibit FLT3. And FLT3 is an enzyme, so that's a target where it was not too hard to make drugs against. And so these are all molecules that are new, that didn't exist when I was in med school. So it's really exciting.
Yeah, it really, in talking with other researchers and stuff like that, the progress from year to year to year to year is just amazing. And a side note, I love all the names. They're fabulous.
There's so many, and I don't like when they change them.
Yeah, I wouldn't either. I'd be like, wait, I just learned
kind
that
of
one.
like, I grew up with the gene being called TEL, and now it's called ETV6, and they're constantly changing. And
oh, that's great. So all of this promising research and all the things that are being discovered and looked at is in looking at the treatment options based on these discoveries. What are you looking at into minimizing, I guess, the long-term side effects that we hear from, we've had patients in our program for years that are still dealing with long-term side effects. And that's a big concern with them, both physical and mental, but that might be a conversation for another day.
And no, Tara, absolutely. Like this is such an important issue in the treatment of children with cancer.
Yeah.
Are the concerns about the impacts of the therapy on long-term survival and side effects, right? And so, when we're treating leukemia or other types of childhood cancer, it is really always that very delicate balance between curing the cancer but trying to minimize side effects. And so, in the case of leukemia and other pediatric cancers, but particularly in leukemia, as we've gotten better and better at this risk stratification, we've been able to back off on some of the more toxic therapies as we now have better information about which leukemias are easier to cure based on the gene changes and which leukemias are harder to cure. So, compared to when I was training, in med school and maybe even earlier in like residency, there are patients that now we no longer use bone marrow transplants for, right? And the bone marrow transplants certainly have a lot of toxicity. And so as we've learned that, okay, this gene fusion, those patients do really well, we don't need to use a bone marrow transplant. And we didn't know that back when I was in med school. Or there are children who have a certain gene change called the BCR-ABL gene change, either in their ALL or they have a different type of leukemia called CML. We now have fantastic drugs that inhibit BCR-ABL. It's an enzyme. It's a fusion that's an enzyme. So again, lots of good drugs against that. So for many of those children, they no longer will need a bone marrow transplant. So they won't have the toxicity of that transplant. So one of the great things is that, we've been able for certain patients to back off as we have better knowledge of what leukemias are easier to treat or harder to treat. And ultimately, of course, what we want to do is to try to eliminate all of the therapies that have these long-term toxicities. We're not quite there yet, but that's the long-term goal, is as we can make more precise drugs that target the cancer and not the normal cells, that's when I think we're going to have, the best long-term cures and the least toxicity. And so that's our goal. And we've certainly made progress from, again, we'll go back to when I was in med school, but there's still more work to do, absolutely more work to do.
For that, yeah, that's it's fascinating.
And the other thing we're doing is, for some of the drugs that have known toxicities, so for example, there's a drug called doxorubicin, another similar drug, daunorubicin, that can be injurious to the heart. We now have other medicines that we can use to try to decrease the impact on the heart. So sometimes, even if we have a medicine that we know can have long-term toxicity, the field has been able to identify additional medicines to try to counteract the toxicity. So that's another strategy we've been using. Of course, we'd like to get rid of it completely.
Right.
But until we have better drugs, another way to mitigate the side effects is by these drugs that counteract the side effects.
Yeah, and kind of look at it from the different angle. Yeah.
Right, exactly.
Yeah. Excellent. So let's talk about how the grant funding received from CLRA has impacted the future of blood cancer.
Yeah, no, so as I said, Tara, we have been just so grateful to the support that CLRA has provided. And, just as I said, the warmth that we have received as a lab in interacting with you all, it's just been, such a nice relationship over the years to work with you. And, when I think about the impact, it comes in different layers. And so one is, the support has helped us to push technologies forward that have led to new discoveries in the field of leukemia. And so some of our earliest genomic screening technologies were supported by you all, and some of the newer strategies that we've been using in kind of the early days of our using CRISPR screening in the lab. So that's one big area. The second is the support helped us validate a number of targets in childhood leukemia. And that validation has now led to efforts to try to find drugs. So the first step, when you find a new target, you have to validate it. And then, the next steps, which are hard, are finding the drugs. And so we now have a number of collaborations with chemists to try to find drugs against those targets. So that was another big area where the support has been so critical. A third is that a number of years ago, we had, as I told you, we had these tests to try to identify gene changes in leukemia. And we were starting to build a growing collection of drugs that target those changes. And so one of the areas where you all helped get a big project off the ground was a study that we did that was a match targeted therapy study, which involved a number of institutions across the country, where we were asking, can we identify gene changes in leukemia? And then are there drugs available that might inhibit those gene changes for children with very highly relapsed refractory leukemia who didn't have other treatment options? And we have some examples from that trial where this strategy, we're bringing the genomic testing to that child's leukemia in the kind of early days of doing this, right? This was a number of years ago, helped us to identify drugs that then had a very significant impact on the care of the child. And so that was a real world example where it had an immediate effect. And to me, that's a highlight like no other, when something that you're doing in the lab has an immediate impact for a child that you're treating, And then I would say, lastly, something that I think a lot about is building the pipeline of the next generation of researchers. And so the support from you also helped the young people in my lab to do their projects. And now these are people that have their own labs that are focused on leukemia, and they're, our next generation. So, it's been really tremendous working with you all.
Yes, we love to hear that. That's great. Well, we are so thankful for your time, and I'm going to let you get back to the lab. And I'm also thankful for Amy and people that work with you who are just amazing and fun to work with, honestly.
Yeah,
And they
it's a
make,
great team.
yeah, it's a great team. They make the process great. So thank you again for being here. And next time I'm coming to see that lab in person.
Awesome. We'd love for you to come. We'll show you around. We'll show you some leukemia cells under the
I
microscope.
would love it.
Yeah, no, we love to have folks come and visit us. So please do, Tara.
Will do. All
Okay.
right. Thanks so much.
Thanks, Tara. Thank you, everyone. We really appreciate it.